And thank you for joining us today for our panel discussion entitled Innovations and Breakthroughs in Gastrointestinal Cancer Trials, Treatments, and Patient Care. So our panel discussion series provides our patients and our supporters an opportunity to hear from our leading physician researchers about innovations in the field of GI oncology. So I've noticed that we're joined today by many familiar participants. I wanted to thank you for your continued interest and support of the GI cancer program here at MGH. Thanks to partners like you, we've been able to challenge the standard of care and bring innovative ideas to benefit patients. For those of you who are new to the series, I want to welcome you, and I'm very appreciative of your time and attention. And we hope you'll be as excited as we are about the progress, hearing some of the progress we made and some of our plans for the future. So before we start, I wanted to go over some general housekeeping items. During this Zoom webinar, you'll be able to see and hear us, but we will not be able to see and hear you, unfortunately. But if you have any questions, please feel free to submit those through the Q&A function at the bottom of the Zoom menu bar. There is also an option, just so you know, if you prefer to submit the question anonymously, you are able to do so. So with that, we look forward to getting going. So as many of you have noticed, this has been a very unusual year, a very challenging year. We faced really, all of us have faced just some unprecedented happenings in the world. And COVID in particular has put some tremendous strain on the hospital, on the cancer center, on our program. During the surge here in Massachusetts back in April, we actually had to transition many of our oncology floors into ICUs and to makeshift COVID units. Many of our oncology colleagues had to actually cover COVID units, work in the ICUs again, and we had to shut our research laboratories down for about 2.5 months. And so this certainly created tremendous logistical pressures, tremendous financial pressures for our research program, and has really just been an unprecedented time. But thank you to, thanks to our partners and your support, we actually have been able to keep our momentum going and have really achieved this year some phenomenal successes in the field of GI cancers. Some of those include, some of you will hear about today, but some of the notable achievements this year include pushing forward one of the first clinical trials combining both targeted therapy and immune therapy for about 10% of colorectal cancers who harbor mutations in the BRAF gene, one of the most lethal molecular subsets of colorectal cancer. And so for the first time, combining both a targeted therapy against the mutated BRAF gene with an immunotherapy meant to unleash the body's own immune system against the cancer, we've actually been able to achieve unprecedented response rates, and importantly, also unprecedented durability and long-term control of these responses, which is truly a meaningful advance. Our team has also made some tremendous contributions, both in the laboratory and in the clinic, on developing new therapies for approximately 10 to 20% of biliary cancer patients that harbor a specific gene rearrangement in a gene called FGFR. These insights are now driving a new wave of therapies, some of which have just been approved earlier this year, and we expect several more of those to be available to patients in the years to come. Another example is that our multidisciplinary team of researchers spanning surgery, radiation oncology, and medical oncology have been pioneering new and innovative ways of delivering therapy pre-surgery for patients with pancreatic cancer and gastric and esophageal cancer, which have been leading to unprecedented rates of successful surgical resection, long-term survival, and even cure in these diseases. And this is becoming a very exciting part of our program. And finally, we're integrating new technologies, including some that you'll hear about today, which we call liquid biopsy or analysis of what we call circulating tumor DNA. And what this involves is essentially now a simple blood test, which we can use to detect the presence of cancer after a patient has had surgery, to determine whether that patient is likely cured by that surgery or whether that patient may harbor hidden tumor cells somewhere in their body that we can't see with conventional methods like CT scans or typical laboratory tests, so that those patients can get therapy to salvage the chance of cure. So we'll hear about a few of these exciting developments today. One of the things I wanted to note about our MGH in general, our cancer center and our program, is that we really believe in bringing people together to accelerate the rate at which we can improve outcomes for patients. So this involves both what we call multidisciplinary care, where doctors and practitioners across multiple disciplines, medical oncology, surgery, radiation oncology, and others will actually come together, take care of patients together, so that patients are offered a full multidisciplinary mode of care to really optimize treatment. But we also bring together laboratory researchers and clinical researchers so that we can make sure that the most cutting edge scientific technology is being applied in the most critical and frontier settings of clinical research. And this is really has enabled our program, with the support of many of you, to achieve some important breakthroughs over the coming years. So I'm joined today by two of my close colleagues and close collaborators, with whom we spend a lot of time conceptualizing and developing new clinical trials. I'm joined by Dr. Ted Hong, who is the director of the Gastrointestinal Service in the Department of Radiation Oncology, and Dr. Aparna Parikh, who is an assistant professor in medical oncology and a member of the Tremere Center for Targeted Therapies. So I would like to ask both Ted and Aparna to share a little bit more about some of the work that they've been doing. So Dr. Hong, do you want to begin?
Thank you, Ryan. And thank you all for coming. It's so nice to see so many patients and friends at this webinar today. Hope everyone had a nice holiday. And for those of you who I haven't seen in person for a while, I really do miss seeing you all. Today, I'm going to talk about how I think about trials in our radiation-based clinical trial, multidisciplinary clinical trial program here at MGH. When we think about our trials and conceptualize what is it we're trying to do in different clinical situations, what we really feel our role here at MGH is to challenge paradigms and to redirect the way that we take care of different diseases. Part of this is a lot of our early phase innovative trials are being done only here at MGH, and in the setting of a single institution, it's really our opportunity to try some more out-of-the-box ideas. Additionally, we like to leverage, as Ryan was referring to, our wealth of research and multidisciplinary team-based science to really capitalize on the wealth of research behind us in the lab to do things that no one else can really have the opportunity to think about, and also leverage our very unique multidisciplinary setting to do the types of trials that really aren't feasible at other institutions. And to demonstrate what I mean by this, I'm going to touch on 3 different clinical scenarios that we're exploring, that we've been doing clinical trials and are exploring future directions in. This includes the refractory setting, meaning when patients have cancer that is no longer responding to standard therapies. Then I'm going to talk about an incurable situation where we've transitioned that scenario to a curable situation, which is ultimately our goal, to cure more patients. And then I'm going to finish with a curable situation where we're trying to better select patients for non-operative management and maintain their quality of life and survivorship. Again, all examples of a multidisciplinary team-based approach to try to transform the way we take care of cancer patients. So let me start with the refractory setting, or what many think of as a hardest setting, where patients have no standard options for their cancers. One approach that we've been working on, I've been working very closely with Dr. Parikh on, is trying to get immunotherapy to work in gastrointestinal cancers where historically it hasn't worked. The 2 most notorious disease situations that this occurs in are colorectal cancer and pancreatic cancer, particularly when they're what we call microsatellite stable, which is the majority of pancreatic and colorectal cancer. This simply means that they are a non-hereditary form of cancer that doesn't have a lot of mutations. And I'm going to get back to that point in a second. So the way that our current immunotherapies work is our immune system has accelerators and brakes. Accelerators, so if we get an infection, be it bacterial or viral, our immune system ramps up, but also brakes, so our immune system doesn't attack our own organs and create autoimmune diseases like rheumatoid arthritis or inflammatory bowel diseases like ulcerative colitis or Crohn's disease. Cancers, as you know, are not normal. So in theory, our immune system should be able to recognize that that they're different. But cancers are also really smart, and they co-opt a system in which they put proteins, think of them as flags, on their surface to signal to our immune system, don't attack me, I'm normal, even though they're not normal. So the way that the current generation of immunotherapies work are that they cover up that interaction between the cancer cell and the immune system, so the immune system can say, aha, you're not normal, I should attack you. And that strategy works great in diseases like melanoma and smoking-related lung cancer, because those cancers have so many mutations that they make a whole bunch of abnormal proteins that are easy for the immune system to recognize. The problem with colorectal cancer and pancreatic cancer is that they're what we call genomically bland, meaning they only have a few mutations. So covering up that signal to say, don't attack me, is insufficient to get your immune system to respond. And that's where radiation comes in. We had an idea that we could get immunotherapy to work in colorectal cancer and pancreatic cancer, where the historical response rate is 0%. So in other words, no responders, by simply adding 3 shots of radiation to one of the cancer spots with the immunotherapy. And we initiated a study back in 2017 that is currently completed and now under review. And we were actually able to show that in about 1/5 of patients, they were able to respond globally to the immunotherapy. This represents by far the highest response rates reported in a clinical trial for both colorectal cancer and pancreatic cancer. So based on this observation, we have now initiated 3 more trials, with other trials coming in behind that are now currently open and accruing to further improve upon this concept. So that's one example of the way that we're thinking to try to get treatments to work in very difficult situations where they don't currently work. I'm going to switch now to another incurable situation, but one that we frequently see at the time of diagnosis, and this is locally advanced pancreatic cancer. I'm sure many of you know, and those of you who have been through pancreatic cancer treatment know, pancreatic cancer is a notoriously difficult to cure cancer. And in a large part, that's because 85% of patients at the time of initial diagnosis do not have a cancer that's amenable to resection, meaning it cannot be removed. And if the cancer cannot be removed, it cannot be cured. Locally advanced pancreatic cancer represents about half of all new diagnoses of pancreatic cancer, and is characterized by the cancer being just in the pancreas, but involving blood vessels that cannot be safely removed. Historically, the results with our treatments for locally advanced pancreatic cancer lead to a survival of less than 1 year, and less than 5% of patients being converted with our treatments from unresectable to resectable. In other words, less than 5% of patients that we typically treat historically have been able to get a curative operation. So based on this, about almost 10 years ago now, we had an idea, a twofold idea. One, there was a new regimen from France called FOLFIRINOX that looked like it had better activity. And we had an idea that if we gave all the chemotherapy and radiation before the operation, we could potentially improve the likelihood of cutting it out. But we did not think that that would be enough. At one of our labs in the Department of Radiation Oncology, they had been doing some work showing that a simple blood pressure medication called losartan, which is an angiotensin receptor blockade, so FDA approved, millions of patients getting it, could actually decrease the amount of scar tissue within a pancreatic tumor, improve chemotherapy delivery, and improve the likelihood that we could shrink the cancer. So back in 2013, we initiated a 50-patient clinical trial where we gave FOLFIRINOX with losartan followed by radiation, and we were able to take out the cancer in over 60% of patients. This is an over 10-fold improvement in the likelihood of having a potentially curative operation. On average, our patients are living well beyond 3 years, and many patients are cured. So this has been transformative, and this has led to our current trial that we had sponsored by Stand Up to Cancer, where we're specifically asking, what does losartan add and what does immunotherapy add based on the studies I was just discussing, to improve the likelihood that we can cure more patients with pancreatic cancer. This idea has become so prevalent and has gained so much scrutiny in our field that I've had to give 5 presentations at other cancer centers in the last 8 weeks on this very specific topic. So this has become the hottest issue in pancreatic cancer. And this development could have only happened at MGH, where we had the science and the multidisciplinary team to pull this together. The last disease I would like to talk about is rectal cancer, another disease that I frequently treat with my colleagues in surgery and medical oncology. Now, we're pretty good at curing rectal cancer, and we cure about 70 to 80% of our patients with rectal cancer. But it comes at the cost of not just going through the chemotherapy and radiation, but also a big surgery that can affect whether or not you can need a permanent ostomy or not, as well as whether or not one develops things like sexual dysfunction or other bowel dysfunction. So the hottest question in rectal cancer is, are there groups of patients in whom we can avoid surgery in? And there's a two-prong approach that our group is thinking about now in order to improve both the selection and the likelihood that someone doesn't need the surgery. One approach is based on evaluating tumors at the time of diagnosis as well as after treatment with radiation and chemotherapy to see what genetic changes and molecular changes occur to point us in a direction of how we can improve the likelihood that we kill the cancer completely without surgery. The other approach that we are looking to initiate is to use a liquid biopsy approach called ctDNA to tell us in which patients is it safe for us to not offer the operation and to avoid needlessly doing the operation, but also avoid misselecting patients to not do the operation who might be at risk for recurrence. Because ultimately, in these highly curative situations, we're really interested in doing things that maximize quality of life. And that may be organ preservation, that may be training of colostomies so that patients can avoid wearing a bag and simply empty out their colon at very specific times. These are the quality of life approaches that we are taking to try to improve outcomes in diseases where we can already cure a lot of patients. And with that, I think this is a good time to transition to Dr. Parikh, who can explain a little bit more about these liquid biopsies that we're hoping will guide our future therapies.
Great, thanks, Dr. Hung and Dr. Corcoran. It's a pleasure to be here. I'm 1 month into my maternity leave with my third kid, so it's nice actually to put on my kind of scientific and clinical hat again and sort of talk in adult language. And I think, like Dr. Hong and Dr. Corcoran highlighted, one of the, I think, greatest assets to the MGH Cancer Center and the GI group is this interdisciplinary work together. I'm not a lab scientist, but over the years have come to really see the clinic as my lab. We're constantly asking questions and learning from patients that we're seeing in front of us to try to move the needle in terms of gastrointestinal cancer patients. And Dr. Corcoran and I work very closely together, really trying to think about novel lab technologies and how we can move them into the clinic. One of these technologies that was already alluded to by both of them was this concept of circulating tumor DNA. And as a very brief 101 of circulating tumor DNA, what it is all of us, our normal cells are shed in the blood. There's DNA that we can detect in the blood. But we can also, in the blood, start to pick up tumor-specific DNA. And with improving technologies, we can pick up tumor-specific DNA to very, very sensitive amounts and are able to pick it up even when there's just a fragment of DNA in the blood. And so what we've done, and back in 2016, with actually a grateful patient, we started a systematic liquid biopsy collection protocol, basically trying to really understand and learn from the blood of gastrointestinal cancer patients. So we basically, every patient that walked into the clinic, we tried to get blood on for circulating tumor DNA. And this spanned the various stages. So this were patients with early stage disease, late stage disease, patients on chemotherapy, targeted therapy, most recently immunotherapy, and trying to start to figure out how we can use this technology in the various settings. So I'll just highlight three areas where we're really thinking about how to use these clinically. The first area is in an area that's called minimal residual disease. So an analogy I like to tell patients when I'm seeing them in clinic is that it takes about 1 billion cells before you actually see residual cancer or a tumor on a CAT scan. So after patients undergo curative surgery for colon cancer, for example, stage 3 colon cancer we'll use as a good place to talk about. 50% of patients are cured with just the surgery alone. 50% of patients have microscopic disease still there, but currently we don't have a way to detect who those patients are that have microscopic disease. So currently everyone after surgery gets chemotherapy, even though 50% of patients are actually cured with just the surgical approach. Then despite chemotherapy, there's still a percentage of patients, around 20% of patients, that will still go on to recur and their cancer will come back. So can we use this technology to better help figure out who the patients are that may not need further chemotherapy and spare them quality of life issues from getting toxic chemotherapy? And are there patients that despite chemotherapy, we know that the patients are at high risk for their cancer recurring? And by looking at the circulating tumor DNA, can we help figure out who those highest risk patients are and give them additional treatment? And with that additional treatment, hopefully improve cure. And this we've actually launched into a clinical trial that's the first actually launched clinical trial in the country, launched back in January, unfortunately, right before COVID. So we did have to shut down briefly, and it's funded by Stand Up to Cancer. And we're doing it with us and 6 other major cancer centers across the country, including Sloan Kettering, and several other, and Hopkins and several other collaborators. And what we're doing is we're looking at those stage 3 patients who are still able to have circulating tumor DNA detected after initial treatment. And based on doing molecular profiling, so actually genotyping the tumor and trying to figure out the genetics of the tumor, try to figure out if we can give them additional treatment to basically what we call clear that circulating tumor DNA, because we think that if we can clear it, that we might be able to actually improve the cure rates. So that's one setting that we are looking at residual disease, but we're also looking at it not just in colon cancer. We have a concept in pancreatic cancer. We're thinking about this in gastric cancer. So it's a technology that can be applied kind of pan GI cancers. The other 2 areas where we've been thinking about circulating tumor DNA a lot is in the setting of resistance. So when you biopsy a lesion, you're only biopsying one particular part of the tumor. And if there's multiple tumors, we are starting to realize that those multiple tumors may all look different if you were to profile them but with just a single snapshot of a tissue. Not only within different, among different tumors, but also within a tumor. So if you had one liver lesion and you were to biopsy in 5 different spots in that liver lesion, they may look a little bit different. So what we need to do is we've learned that with blood biopsies, it can actually capture the tumor genomics holistically and maybe a better representation of what's happening with the tumor at any given point in time. And what we're learning is that we can start following patients over time and we can see in the blood if resistance is happening. And as resistance is happening, we are starting to model these resistance mechanisms and are able to design new therapies to overcome these resistance resistance mechanisms. There was a great example with the FGFR story that Dr. Corcoran alluded to at the beginning. In cholangiocarcinoma, where this approach looking at resistance mechanisms in blood and designing 2nd and 3rd generation FGFR inhibitors really moved the needle for these patients. We're doing that in colon cancer with targeted therapy, anti-EGFR therapy for colon cancer patients. And again, able to figure out when targeted therapy is appropriate and not appropriate. So we're actually using those technologies in real time and in clinic currently. The last area for the blood biopsies is still under investigation, but we're starting to realize that perhaps the blood can tell us if patients are responding or not responding, and tell us a lot earlier than we would know with just a CAT scan. So we did a study of over 100 patients, and we showed that circulating tumor DNA can actually predict response to treatment quite accurately. And so what if we can get a better sense of how patients are doing with just a simple blood draw, and spare toxicity for ineffective therapies, and then keep people on therapies that we know are working, even if a CAT scan is sometimes inconclusive. So there's a lot in that space, but I think it's one of the things that we're most excited about because it's a non-invasive test and it has so many different applications that can really hopefully move the needle for patients going forward. So with that, I'll turn it back to Dr. Corcoran.
Excellent. So thank you, Dr. Hahn. Thank you, Dr. Parikh. So with that, I want to open up this session to questions from the audience. Again, just as a reminder to click the Q&A button on the bottom menu bar. And if you'd like to ask a question anonymously, there is a feature that you're able to do. We do have a few questions to start with. So the first question is that someone remarked that they've seen in other disease areas like lung and melanoma that they've been able to achieve very dramatic success with immunotherapies. Can each of you give a brief answer on how immunotherapies are being incorporated into GI, where we've had less success, and kind of where we think the big opportunities are to really expand the impact? Maybe we can start with you, Dr. Parikh.
Yeah, so I think to Dr. Hong briefly alluded to some of these concepts. So we're basically trying to figure out ways to mount an immune response in these tumors that otherwise are called what we call cold. So radiation is one such technique that we are using to hopefully mount these immune responses. But our group, some work with Dr. David Ting, who's looking at another mechanism of trying to elicit immune responses, looking at what's called kind of non-coding RNA repeats as an example. And these are drugs that are actually used for HIV and hepatitis and things. And we're very keen on approaches such as these and radiation and several different approaches to try to get immunotherapy to work in these settings where otherwise they haven't worked. And I think what's remarkable about it is, everyone is trying to do this in the field, but what's really amazing about kind of our group is all of the work that we're doing is kind of coming from in-house ideas and in-house lab work. Dr. Corcoran's trial looking at targeted therapy and immunotherapy came from work that started now probably 10 years ago with kind of small steps with just BRAF patients. And as we learned more from biopsies and tissue collections and blood, started to think about how we can add on approaches to get these therapies to work. And so, it's a longitudinal kind of, it's a marathon, right? It's not a sprint for this. And we learn from every trial, we learn from every patient, and we're learning kind of iteratively from our own lab work and our colleagues kind of in-house of what the best mechanisms are going to be to try to get these therapies to work for patients.
Thank you. And Dr. Hahn, did you have anything you wanted to add, or that was a very comprehensive answer?
Yeah, I would just add that, as Dr. Parikh was alluding to, there's a lot of complexity in understanding why an immunotherapy doesn't work. And I sort of alluded to a simple reason that a lot of the cancers that we take care of just don't have that many mutations, so they're just not that funny looking, so to speak. But there's a lot that also involves what we call the tumor microenvironment. This is the area around the cancer cells themselves that may have either different types of tissue, or they may host different types of immune cells, some of which may be actually immunosuppressive. So a lot of what we're trying to do is really tease out on a single cell level exactly what is in that environment and what can we alter then in the future to shift the area to being a more immunogenic environment. So along those lines, in many of our clinical trials, we are doing repeat biopsies with very intensive analysis with techniques like whole exome sequencing and single cell RNA sequencing, single nucleus RNA sequencing, in order to really understand what sort of pressures and shifts are happening at the single cell level that can hopefully provide us a window to getting these therapies to work better. The way I always think about it is that we've opened the door with some of our approaches, but we now need to throw the door wide open. And I think that these sort of intensive investigations into the tumors that we treat on these studies is really key for us to think about the next generation of trials.
Yeah, so thank you, Dr. Hung. So I think this was really a fantastic question for whoever sent it in. Thank you. I think that what we've seen in other diseases is that a very, very when immunotherapy works, it can provide very long-term disease control, and in some cases even curative disease control. And unfortunately, those same therapies have just not panned out in GI cancer. And I think really one of our biggest challenges, and something that we've directed a tremendous effort against, is that we're really attacking this problem from multiple angles to try to design ways to really expand the benefit of immunotherapy. And so both with efforts against radiation and others that you've heard, I'll make one other quick point to something that Dr. Parikh mentioned, that we've actually found some very encouraging data that actually some of the very targeted therapies we use, which actually directly block and inhibit the actual mutations, gene mutations that drive a certain patient's cancer, that many of those can actually cooperate to actually make those tumors more responsive to immunotherapy. And we've actually been able to study this really through an unprecedented collaboration between the clinic and the laboratory, where we've actually created an entire facility in the hospital where tumor biopsies can go directly upstairs from the interventional radiology suite and undergo techniques such as Dr. Hung mentioned, like single cell RNA sequencing, where we literally can dissociate the tumor into tens of thousands of cells, some tumor cells, some immune cells, some normal connective tissue cells, and figure out exactly how each of those cells are changing in response to treatment. And this is really actually showing us how these, the potential to actually link two of the most revolutionary therapies in the last decade or two, targeted therapy and immunotherapy, how we can actually rationally link those to make new responses. I think, again, kind of an example of some of the very unique efforts that we're able to achieve here through the collaborative system that we've built between the lab and the clinic. So I have another question. I might direct this one to Dr. Parikh. This person says, I saw that during the pandemic surge, the cancer center physicians and nurses were redeployed to help meet demand. How did the hospital shutdown impact your research program?
Yeah, so many of us were kind of redeployed. I think the term redeployed is a little bit tricky because, I think we didn't kind of leave our cancer hats. We did spend some time on the COVID floors, myself included, but very much as we could try to, we followed our patients and saw patients in clinic, and then tried to push forward the research agenda and program as best as we could. So the way we did it was by disease group and by cancer center, really a thoughtful and hard look at every research protocol and the resources involved, and what research protocols, if we felt like there was no other option for patients, those are programs that we tried to really keep open as best as we could. We moved to a lot of I hope some of these things that we moved to will actually stay, like remote monitoring. So not having patients having to come back into the clinic, local blood draws. Some of these actually liquid biopsy companies and things were actually setting up mobile home draws at home to be able to send a team of people to the house to draw the liquid biopsy, and we'd get the results, and it would obviate a CAT scan, for example, obviate the need for patients to come downtown. So it wasn't like everything shut down entirely, but there was a very hard and thoughtful and thorough look at every research ongoing protocol that was open, and which ones we felt were absolutely essential for our patients to keep open, and then other ones where we didn't feel were as critical and there were other options for patients that could tide them over until we got through the surge. Those we did shut down temporarily, but we just right before I went on leave, we were looking at our numbers in the Tamir Center as well as the GI group, and we bounced back pretty quickly in terms of being able to get people back on studies. So, and we did take a hit, but I think we kind of came through that surge quite well, actually, considering what we all went through.
Thank you. And there was no question that it was kind of an incredible and unprecedented disruption, but it really was inspiring to see how all the members of the team throughout the GI Cancer Center, the Cancer Center as a large, and the hospital really banded together to continue patient care, even when sometimes we couldn't see patients physically in the same room, which is a very new thing we're getting used to, and to really keep the research program. It's amazing. I think it's amazing what we were able to accomplish this year despite this unprecedented challenge. So I have a question now that I'm going to throw toward Dr. Hong, again from one of our audience members. Have there been any thoughts about using circulating tumor DNA to diagnose colorectal cancer before more invasive testing?
Yeah, I think that's another great question, because ultimately, the best way for us to cure cancers is to catch them early. And if you look at the things that really drive the improvement in cure rates of cancers across the board, that has been most pronounced in areas where we've had successful screening procedures. So yes, absolutely. There's a lot of interest in using these liquid biopsy technologies in the setting of early detection. A couple points that I just want to bring up regarding some of the subtleties of the way that we think about it and why it's not so easy as to say we have a technology and we can just apply it to that situation, but what our group is very actively thinking about. First of all, there is a number of different types of ctDNA technologies, and some of them may be better suited for evaluating why a patient's tumor may not be responding to Dr. Corcoran's latest and greatest treatment, and is there something else that he can think about to alter that treatment? Another use that we're very interested in is, are there patients after they get a potentially curative treatment with surgery, chemotherapy, and radiation, are there a group of patients at a higher risk who we need to think about maybe doing more because they still have some of the ctDNA? And that might be a different technology. The analogy that I use in that regard is a little bit akin to many of you have gone through imaging before. And when we say imaging, you know that means a picture, but sometimes you'll hear me say we need to get a PET scan. Sometimes you'll hear me say we need to get a CT scan. And sometimes you'll hear me say we need to get an MRI. And those are all different. Along those lines, I think that what we are trying to figure out is which specific technology is going to be the most useful in achieving two things in the early detection setting. One, having what we call the sensitivity to pick up something early, so not missing a cancer. But two, also having the specificity, meaning making sure that if we see something, we're not going to have to jump and overreact in some way. Because a lot of precancerous lesions in the colon, like polyps, actually have early mutational changes that are common within cancers. So decades ago, Dr. Vogelstein at Johns Hopkins had outlined this 10-year process where colon cells can accumulate mutations on their way to becoming polyps and then invasive cancers. All a way of me saying that a lot of non-cancers can have alterations that are common in cancers. So I agree that yes, we are very interested in applying this technology to the early detection setting, but our group is working very hard to try to tease out some of those complexities.
Great, thank you, Dr. Hong. So I have another question, an anonymous question. Again, just as a reminder, if you want to ask a question anonymously, it's very easy to do so. For Dr. Parikh. So this person would like you to explain more about how you model cancers when you're following patients over time with circulating tumor DNA.
Yeah, so I'm not, and feel free to type in more if I'm not quite getting at what you're asking with the model, but I can sort of tell you a little bit how these liquid biopsy technologies work, and I hope that'll get to your questions. So And the technologies are a little bit different based on the setting. So it's a little bit different if you're looking at it in a patient that has metastatic disease, for example, versus the technologies that we use if a patient, when we're trying to do it for early detection, for example, or in the residual disease setting post-surgery to see if there's anything left behind. So in the metastatic setting, what we're able to do now is we're able to do these broad molecular profiling the same way that many of you may have had your tissue profiled by either our MGH snapshot or Foundation Medicine or another platform. We're able to now run those same next generation sequencing on the blood. And what I was alluding to before is that with the blood, we're able to better capture the heterogeneity of the tumor. And so we're able to get that genomic information now, not just from the tumor, but from the blood. And so you can do these large, 400 gene panels now in the blood and detect these alterations that you would in the tissue. And by having it in the blood, you can do that over time. And so the modeling is, I think the beauty of the blood biopsy is that it obviates the need necessarily for repeat tissue testing at any point in time, because you can just get a blood draw and you can follow what is happening to different mutations over time. And so you can see if there's new mutations that are arising that may be actionable or targetable, or new mutations arising that would confer a therapy no longer to work. And so I think the modeling is really that kind of spatial, not just spatial modeling, but the temporal modeling, I should say, kind of over time to see what's happening with patients. And in the residual disease and kind of early detection setting, we aren't able, we want to capitalize on sensitivity. So we want to really try to be able to detect anything that we can see that may be cancer derived. And so we don't necessarily go as broad on the sequencing panels as we otherwise would in the metastatic setting, but we have kind of customized panels that have been designed to capture the bulk of mutations that you might see in an early stage colon cancer, for example. And what we're learning is that cancer versus normal tissues have different patterns of kind of genes being expressed, that we can, not just with the genes itself, but the way they're turned on and turned off, there we can look at these in the blood as well. It's a technology called methylation, and the DNA, that how the DNA is fragmented, kind of broken up, is quite different in cancer cells versus normal DNA. And so these are all approaches that we're using to try to detect with specificity what is cancer and what is not cancer in the blood.
Great, thank you, Dr. Parikh. And I think, as we can tell, we've spent a lot of time talking about liquid biopsy and circulating tumor DNA today. This is an area that we've made a huge investment in, both in time and effort and resources over the last 4 or 5 years in a variety of areas that we've discussed today: early cancer detection of residual cancer after a curative surgery, real-time tracking and monitoring the actual evolution of the tumor in real time, so that we can actually use that information to try and figure out how we can better design therapies to prevent cancers from becoming resistant. And this is really something that we think is going to be a transformative approach on multiple levels, and probably, I would say, something that we've turned into probably one of the largest liquid biopsy operations, at least in GI cancer, perhaps in the world. And it's remarkable that this effort really would never have happened, and is only possible because of some support from our generous supporters, some of whom I believe are joining us today. And so this is really, I think, going to be an exciting field to watch. So I have another question that was sent in. It's actually a practical question that I'm sure is coming up for many patients. So for a patient currently on immunotherapy, the question is, will they be able to get a COVID vaccine? And I know that's always something that's best discussed directly with your personal oncologist, because there are nuances and scenarios. But any information we can offer as a general advice?
Yeah, I can take that. And I'll use the flu vaccine anecdote actually a little bit as a segue into this discussion. Obviously we know that cancer patients are at higher risk for developing serious infections from COVID than someone without cancer. So, cancer patients, and this is coming up in our division as well, will be prioritized at some point in the vaccine. I think priority should be used carefully right now because obviously the state is determining priorities for patients, and then or for people, the population level, and then it'll be kind of up to the hospital and cancer center to figure out how to disseminate into whom. But I think, part of it, like Dr. Corcoran said, will be a discussion. But, with the flu vaccine, for example, and immunotherapy first kind of came to the stage for all patients, melanoma patients, lung cancer patients. There was some concern that maybe the flu vaccine was perhaps there were some more side effects for the flu vaccine in patients with immunotherapy. And so some of the earlier trials, even that we did, the flu vaccine was precluded for patients on some of these studies. And but what we learned with just following patients over time was that the flu vaccine actually was not detrimental for patients on immunotherapy, and now everyone is getting the flu vaccine on immunotherapy. And so I know it's not a very satisfying answer, but I think, we'll be prioritizing cancer patients, and part of it will just be learning and kind of research and following patients over time and carefully to make sure that we're not doing any harm. I think all of us operate under the dogma of first do no harm. And if the imminent threat is COVID, we all feel that cancer patients should be prioritized, but it will take, us following patients over time to make sure that they're safe. And as we're doing that, we're going to be studying how patients are doing as they get the vaccines and things too. So I wish I had a more definitive answer, but I think with COVID in general, none of us have a lot of definitive answers. So it's going to be iterative, and but I can assure you we're thinking about it. I don't know if Dr. Hong, if you have anything else to add to that, or Dr. Corcoran.
No, thank you. I think that's
Yeah,
right. I think
it.
that that hopefully this is something that we all anticipate will be available to us very soon, and that encourage all of you to speak directly to your oncologist about it, so that we can help the world get back, and all of our lives get back to some semblance of normal. So there's another question which I can actually take, although if anyone else wants to make a comment. So many of us have mutations in a gene called KRAS, and for a long time there were no therapies for this specific gene. I've heard there are new inhibitors against KRAS. Can you explain how these are influencing patient care? So I can maybe start this off. So many of you may have heard this term before, the name of this gene, and the reason why is because it's actually mutated in about 20% of all human cancers, not just GI cancers, all cancers. So 1 out of 5 cancer patients has a mutation in this gene, and it really represents almost the master hub of cancer driving signals. And because of the nature of what the KRAS protein looks like, it's been incredibly difficult to actually design a drug to inhibit this. And so for probably about 3 decades, academics and drug companies alike have tried and failed to drug this. But actually just in the last few years, we have seen a new wave of inhibitors, that actually can now target KRAS, mutant KRAS directly and shut it down. And this is, I think, something that really is going to be a game changer for all cancer patients, but particularly for patients with GI cancers, and something that has really become a core of a large research effort here at MGH. So just to give you an example, 40% or 40 to 50% of colorectal cancer patients harbor RAS mutations, and about 90% of pancreatic cancers harbor KRAS mutations. So this is a huge population. And what we're now doing on many fronts is to actually to study how we can deploy these inhibitors in various cancer types. We've really taken our entire, what we call our translational research platform, which is research that spans the clinic to the laboratory, and brought everything we have to bear on this topic. These include patient-derived models that we can derive from patients' biopsies, our liquid biopsy platform that we've talked about today, our genomics platform, and have really created a full-bore kind of assault to see how we can most rapidly and most effectively figure out when and why these inhibitors will work best, and for those patients in whom they're not working, what other therapies we can combine them with to improve outcome. And we've already made some very important research observations that are now being tested in a new wave of clinical trials. It's very exciting. One of the other things we're doing is in another collaboration between the clinic and the lab is actually modeling how targeting KRAS may actually enhance the immune response and make those tumors more responsive. And so we've collaborated with laboratory and clinical and translational investigators to really deploy this. And we think this will be just an incredibly exciting opportunity for our patients going forward. So, if there are any more questions, please go ahead and send them in. I think, one of the things that we focused on is, I know we've talked about a number of things today that were really made possible through the support of our donors and our friends. I wanted to ask each of you, Dr. Hong and Dr. Parikh, if you could give a specific example of how philanthropic support has really allowed you to take on an innovative challenge or make a research contribution that otherwise would not have come to pass.
Sure. So why don't I start? So the first story that I told was about trying to get immunotherapy to work and using pancreatic cancer and colorectal cancer and using radiation in order to make that happen. Now, that idea, when we first thought of it back in 2017, was an idea that there's no real patient experience with it. So getting a traditional grant like from the NCI would have been very difficult. And when we spoke with the pharmaceutical companies that make the drugs about potential clinical trial support, they thought that it was too risky to do. So it was actually, that initial study was actually funded by a grateful patient. So that entire feasibility of being able to do that first study was 100% sponsored by philanthropy. And what that yielded, what that initial investment yielded, besides the excitement that we actually got it to work, was we subsequently got 3 trials then funded at $1 million each. So that one initial philanthropic gift that gave patients options and allowed us to try a new therapy also allowed us to open 3 successor trials and build a whole program out of it. So that seed money ended up being completely vital to the growth of this program now that the whole country is looking at in this combination immunotherapy and radiation for GI cancers.
Yeah, and I think to add to that, I think what a lot of the philanthropy money does is start some of the seed ideas that lend themselves later to kind of larger support, even from pharmaceutical companies. Some of the drugs that we use. One of the trials that I did with Dr. Tang, looking at a drug again that's used for HIV and hepatitis, that we're now moving forward with some other concepts. That drug is a generic drug. And so no pharmaceutical company was really interested in funding that because there was no, kind of market for them for that drug. But now that we had some kind of early interesting data from the biopsies and things and have a clear path of how those may combine with kind of drugs that are there now being developed by different companies, that initial kind of philanthropic organization or funder that gave us the seed money for that trial has now lent itself to other concepts. Same with the blood-based protocol. We did this initial work looking at response prediction kind of from philanthropy funding, and that enabled us to collect serial blood on patients that could tell us if we're able to predict responses. And with that kind of seed money, was able to apply for not only another NIH grant, but now we have some major companies in the blood-based diagnostic space that are funding a larger trial right now for being able to predict early response with blood-based biomarkers. And so I can't echo kind of Dr. Hong's sentiments enough. A lot of the things that we do, because we are in this era of companies and things that do have to look at their kind of bottom line. They may not be as interested initially, but when we're able to show with some early data, which often comes from kind of grateful patients or foundations and philanthropy, that we are actually making tremendous strides in these areas, then we pique their interest, and then the money kind of rolls in rapidly thereafter. So.
Fantastic. So I think I think we're coming up on the end of our time this morning. So I just wanted to thank everyone once again for joining us in the session, for the great questions. We hope you found it exciting. And thank you all again for your continued support of the MGH Cancer Center and the GI Cancer program. You know, if there are any more questions that we didn't have time to get to today, I want you to know that you're always welcome to reach out to me directly by email. I'm happy to answer them myself, or connect you with one of our colleagues who's best able to answer it. So, you know, I made a comment at the beginning about how the culture of teamwork is really just integral to our entire approach to battling cancer at MGH. And this includes both teamwork between caregivers from multiple disciplines: surgery, medical oncology, radiation oncology. Also teamwork between laboratory scientists, clinical scientists, teamwork between physicians and caregivers and their patients, but also teamwork between our supporters. One point I'll make is that numerous times today we saw that a lot of some of the most exciting breakthroughs and advances and the most exciting efforts that we have going really would not be possible without philanthropy. And one of our goals here is to really not just to kind of address basic questions, but to really challenge the standard of care and to bring kind of the unparalleled science at MGH and the unparalleled clinical operations, and really bring them together to accelerate breakthroughs for our patients. And many of the most exciting approaches, a lot of our biggest successes, you know, at the time that they started, they were deemed, you know, too high risk for standard federal funding. And so really many of these things really would not have happened without your support in the past. And I think, as we all know, we've been you know, pleased by the progress we've made this year, despite some of the unprecedented challenges posed by COVID. But this year, more than ever, we're counting on your continued philanthropic support to allow us to keep continuing to catalyze these important breakthroughs for our patient. We hope that you will consider continuing to partner with us for that. So with that, I'd like to thank you all for your time this morning. Thank you all for attending, and I wish you all the best.